Hello, welcome to this talk. I am Maarten and I'm pleased to present our multifocal study of locally advanced prostate cancer. The vast majority of cancers that end fatally are due to a process called metastasis, where cancer cells can disseminate throughout the whole body to other tissues. Therefore, it is of fundamental importance to understand the difference between primary tumors that can metastasize and tumors that cannot. However, in practice, it is not straightforward to clearly delineate these two groups. One relatively reliable way to do this is to use the scores provided by pathologists based on lymph node resections. And the N0 group depicted here is a group that does not contain any cancer cells, as far as the pathologist can tell, in their lymph nodes, while the N1 group contains tumor cells in the lymph node, and here we can say that the tumor cells are capable to metastasize to the lymph nodes. In this setting, we can use a sort of differential testing between the two groups, where, for instance, we can compare expression or mutations in certain genes to identify markers of metastasis. For instance, in TCGA, this information is available, and numerous studies have shown that there are transcriptomic differences between these two groups. In prostate cancer, an important complication is the fact that the tumor is often multifocal. And this makes it hard for pathologists to decide where to puncture the tumor in order to perform sequencing. You can imagine that puncturing the wrong place in the tumor can have a profound impact on downstream analysis, as you risk missing signals that are important for metastasis. To investigate the impact of this heterogeneity on metastatic processes, we have performed a multifocal study where per patient 1 normal prostate sample and on average 5 different primary tumor samples were sequenced, as well as some metastatic lymph nodes. As the number of patients is relatively limited, we have opted to perform an analysis at the signature level rather than on the level of individual genes. The signature is hereby defined as a set of genes for which we know what function these genes collectively perform. And we also have assigned each of these signatures to a hallmark of cancer, and the number of signatures for each hallmark is indicated in the figure. The next challenge was to define signature activity based on the expression of the individual genes of that signature. Rather than trying to summarize the signature activity in one number, we used centroids to capture an average expression state in each of the 3 tissue types: normal prostate tissue, primary tumor tissue, and metastatic lymph node tissue. For any new sample, we can then classify it into one of these 3 classes by assigning it to the class with the nearest centroid. And we found that a distance measure based on Spearman correlation works extremely well. Using cross-validation, we could then identify the signatures that could distinguish very well between these 3 tissue types. And as this implies that these signatures change from normal to primary and from primary to lymph node meta, we hoped that these signatures would capture tumor evolution. An example of what we mean by tumor evolution is shown in this figure, where you can clearly see, this is just a simple PCA plot, where you can see that the first component nicely captures the transition from normal, which are the dots, to the crosses, which are primary tissue, to the squares, which are lymph nodes. And we found that especially invasion-related signatures tended to show this behavior. Of course, if we're really looking at tumor evolution in this slide, we need to make sure that the crosses that are closer to the lymph nodes actually are, in some sense, more metastatic than the other primary samples shown in this plot. And to do so, we turn to TCGA, where we also have patients that have no lymph node metastasis. And for instance, for the focal adhesion signature, we could show that indeed patients that have lymph node metas, their primary tumors are closer to the centroid defined on our in-house cohort, which is shown in this figure. And this turns out to be actually significant, and is significant for 38 different signatures. We can draw two important conclusions from these results, namely that the metastatic lymph node centroid contains some information that is related to this invasion process, and secondly, that some alterations that are apparent in this metastatic lymph node centroid can already be detected in the primary tumor. So, based on these two observations, we wondered, can the metastatic lymph node centroids actually be used to identify the most metastatic primary lesion in our own cohort? To identify the most metastatic primary lesion in each patient, we used the voting approach. Each of the 38 signatures identified on TCGA could per patient vote for the primary lesion that resembled most the metastatic lymph node samples. And this was done by calculating the distance to the metastatic lymph node centroid of that signature. Then, by summing these votes across 38 different signatures, we could identify the primary lesions that are metastatic-like across a wide spectrum of different processes. We hypothesized that these lesions were involved in metastatic seeding. To validate that the seeding lesions we identified were indeed related to metastasis, we derived somatic variants from RNA-seq and identified the primary lesions that shared the most somatic variants with the metastatic samples, as indicated on this figure, where the thickness of the line corresponds to how many somatic variants are shared. In 16 patients, we could compare the seeding lesion derived from somatic variants with that derived from the voting scheme introduced earlier, and we found that they agreed in 10 out of 16 patients, which is far more than expected by chance. As a second validation, we ran the PAM50 classifier on every primary lesion in our cohort. It is well known that the luminal B subtype is related to invasive disease, and we could confirm this on TCGA as well. The figure shows the PAM50 subtypes for each of the primary lesions. Predicted seeding lesions are indicated in full black, and you can see that the seeding lesions are luminal B in 7 out of the 9 patients that have a luminal B subtype. This again shows that the lesions we've identified are related to invasive disease. What is striking on the other hand is that there is a poor correspondence with the Gleason score assigned by pathologists, and this is visible on the y-axis. In summary, these two strategies allowed us to confirm that the seeding lesions are related to invasive disease. Having confirmed that the predicted seeding lesions are indeed related to invasion, we wondered if we could identify a common molecular fingerprint for these lesions. Here too, we checked how many times a signature correctly indicated the seeding lesion in a patient. We have taken into account that different patients have a different number of primary lesions. For instance, under random guessing, the probability of correctly indicating the seeding lesion in patient 1 will be 1 out of 5, while it will be 1 out of 2 in patient 17. We can obtain a p-value for each signature by modeling the number of correctly identified seeding lesions using a Poisson binomial distribution. This table shows the highest scoring signatures. Remark that to identify these signatures, we have only compared between samples from the same patient, removing one of the strongest confounders in cancer research. This allows to identify signatures such as oxidative phosphorylation that are well described in literature but cannot be found in TCGA, presumably because the basal metabolic state is already different between different cancer patients. I hope you have enjoyed this presentation. For more information, please reach out to me, either by email or in person at a conference. Looking forward to it.